Great. Thank you for having me. Thanks to the organizers for putting this awesome mini symposium together and the great resources. I'm excited to kick off some of the science. As I said, I just started my lab just over a year ago at UNC Greensboro, and I'm going to talk today mostly about some of the work that emerged from my postdoc, but also some of the new directions that we're taking the lab. And I'm going to be introducing a term that I didn't use quite so often in everyday life, but I've come to rely on it, a grommet, later on in the talk, and you'll see how that plays into membrane dynamics. Um, so my group is fascinated how cells are constantly maintaining and establishing compartments. And as a group, we're going to focus mainly on membrane-bound compartments, which is, of course, the excitement of this particular group. But we appreciate other compartments as well, other mechanisms of maintaining compartments, which I'll get into. So I'm going to spend most of my time focusing on the a story that we've uncovered of the maintenance and sealing of the mitotic nuclear compartment in the model organism that I study, and that is in fission yeast. So first, a little bit of an introduction to the way that I've come to think about this particular membrane-bound compartment, the nucleus. Of course, it is the mechanism by which we compartmentalize the genome and we segregate the cytoplasm from the nucleoplasm. And we primarily think of the nuclear envelope as a double membrane system, so a lipid-based means of maintaining compartmentalization of two membranes. But I also want to not forget that embedded in the nuclear envelope are, of course, nuclear pore complexes. And nuclear pore complexes are, by definition, a proteinaceous diffusion barrier. They're going to be the second main mechanism that the cell is able to restrict free diffusion to the nucleoplasm from the cytoplasm and vice versa. So this is the basal state, if you will, that we're dealing with in interphase. But of course, this must be dynamically remodeled as we go through mitosis. And as you're likely aware, there are diverse strategies for dealing with how do we access the nucleoplasm and the chromatin inside? How do we manipulate this membrane in order for that to happen? And in the model yeast that I study, as I've introduced as POMBI, they use a quote unquote closed mitosis to solve this problem of a nuclear envelope. So the interphase landscape in fission yeast is going to look very similar to that stereotypical eukaryote that I had, a eukaryotic cell, right? We have a compartmentalized nuclear envelope. But during mitosis, we have to allow a mitotic spindle to be constructed. And what you'll notice from what I've diagrammed here, color coding, is that there is not a loss of nuclear compartmentalization, unlike the metazoan open mitosis. POMBI have evolved a closed mitosis, but they still have to deal with that problem. How do you get your cytosolic cytoskeletal machinery access to the chromatin? And so the way that fission yeast deal with this is that they have a microtubule organizing center, the spindle pole body, or SPB, which is transiently inserted into the nuclear envelope. A fenestra will form in the nuclear envelope membranes to allow for this spindle pole body to insert, create a mitotic spindle. Once you've segregated chromatin, and at the end of mitosis, you will then have a nuclear envelope that needs to be sealed. And so when I started my postdoc, I found this really fascinating because it really introduced kind of two questions to me. So how do POMBI maintain their mitotic nuclear compartment throughout, despite this large hole, hundreds of nanometers in the membranes? And how do they seal this compartment after mitosis? And of course, this is not just a problem that is unique to fission yeast, right? So this is the ultrastructural landscape that has been known for some time now about this opening in the nuclear envelope that has been observed in fission yeast, where we have the spindle body seen here by electron microscopy, the microtubules, the nuclear envelope I'm showing you here in blue. And this is in HeLa cells. This is in telophase, a very similar landscape as sheets of ER come to the chromatin disc to try and reform the nuclear envelope. You have to deal with this same problem. It's just much more tractable, I would argue, in fission yeast to look at this because we only have one singular hole per daughter nucleus that needs to be sealed. And so we thought about this first in terms of a sealing problem, but also in terms of a compartmentalization problem as well. And so when we looked at what potential machinery could be involved in sealing this, the first machinery that stood out to us, as is likely familiar to many of you, is the escort family of proteins. Canonically associated with endosome, multivesicular body formation, interluminal vesicle formation, excuse me, but now well appreciated to be active at membranes across the cell, including at the nucleus, right? And so we thought that this was the perfect opportunity perhaps to implicate these escorts in this sealing reaction. And so being microscopists and being the theme of this talk, we took a microscopy approach and did some live cell imaging of escort machinery at this mitotic nuclear envelope as sealing was occurring. And that's what I'm going to be showing you here on the right. So on the left, I have kind of a model of mitosis in physogenies, just to give you an idea that basically progression through mitosis correlates with the lengthening of a mitotic spindle, and that's shown here in magenta with fluorescently labeled alpha-tubulin. And here I have the linchpin of the escort machinery at the nuclear envelope, or I should say, what's thought to be the linchpin of the escort machinery, and that is the unique nuclear envelope escort CHIMP7. And as I play this movie here, you will see as the spindle elongates, where I have an arrow at the top here, you will see where there's that hole in the nuclear envelope, we get this punctate recruitment of CHIMP7, this linchpin nuclear envelope escort, at around the time when the spindle body is extruded from the nuclear envelope. So we went on to do an extensive array of live cell imaging. I'm just going to show you kind of how we did that here. These are still images arranged in kind of a pseudo timeline going throughout mitosis, starting with early anaphase B on the left, going through actually into the next cell cycle onto the right here. And I hope you can appreciate the punctate recruitment of this particular escort. Now, as you may or may not be aware, the escort machinery is a whole host of, in yeast, half a dozen, in metazoans, about a dozen subunits that are involved in membrane remodeling. And so I'm just going to splash this slide up here to give you an idea of the different escorts that we looked at to get an idea of when they were recruited. The main thing I want you to take away from this slide is that, again, we were able to arrange these by the length of mitotic spindle and other morphological features to get an idea of when these different subunits are recruited. And so the takeaway from this part of our work was basically that we have an initial wave of this early escort machinery, CHIMP7, followed by a second wave that occurred a little bit later, but still prior to the end of mitosis. And then finally, a third wave of different escort subunits. I'm not going to ask you to remember all of these different subunits. It's a bit of an alphabet soup, as folks in the escort field know, especially across species. But I'm happy to talk about that more in depth if anyone has questions about particular subunit dynamics. But basically, the takeaway I want you to have is that there's multiple waves here starting in mitosis, and actually the third and final wave comes after mitosis, and all of these escorts are gone by the following interphase of the next cell cycle. I want to note that all of this required CHIMP7. When we knock out CHIMP7, we don't get any escort recruitment to the nuclear envelope, as has been suggested by work in other organisms. And so this really set up an interesting landscape of we have this membrane remodeling machinery at the right place at the right time to be sealing a nuclear envelope hole. And so that was our next task to look at, are they actually required for sealing? And while we love live cell microscopy, we also love to be able to visualize the membrane directly. And so we took a electron microscopy approach. And that's what I'm showing you here. We actually use correlative light and electron microscopy primarily as a screening tool to allow us to identify from an asynchronous population the timing of a particular cell that we've imaged by fluorescence microscopy. When we image it in electron microscopy, we already know what state of the cell cycle it is. And here on the left, I'm showing you the wild type scenario and interphase cells. And on the right, I'm showing you CHIMP7 knockout cells, where, as I mentioned, we don't get any escort recruitment to the nuclear envelope. And what we found striking is that we saw in all of the interphase cells that we looked at lacking escort machinery, we see this hole in the nuclear envelope, a very small hole kind of opposing the spindle body. And this was really striking to us. I should note that in POMB, there is no lethality associated with Shim7 knockout. The cells are perfectly fine. But when we do the electron microscopy and actually directly visualize the membrane, we can see that their nuclear envelope is not fully sealed. And so this really drove home to us that this escort-mediated machinery is responsible for nuclear envelope sealing. And so that was perhaps intuitive based on what we know about the escort machinery, but we also wondered why, if they're involved in sealing, are they recruited so early in mitosis where you actually have a mitotic spindle and you can't actually seal? And so we turned our attention to our electron microscopy data set where we actually caught mitotic cells. And on the left, I'm showing you wild type again, and on the right, I'm showing you Chim7 knockout. And this time, I'm showing you in late anaphase B. And so in our wild type population, at this time point, we have tight constriction of the nuclear envelope along whatever remains of the spindle. Here, there's no more than one or two microtubules, maybe one that's still passing through the nuclear envelope. So we're relatively late in mitosis. And when we compare that same time point to the Chim7 knockout cells, what you can see is there's this extreme dilation in the nuclear envelope, and that's quantified over here. We only see this in late anaphase B, once the spindle whole body has been extruded and is no longer embedded in the nuclear envelope. And we see here that about a 4 to 5-fold dilation in the size of this nuclear envelope, suggesting to us that perhaps the escort machinery prior to sealing is serving an additional function, and that is to maintain a tight constriction around the nuclear envelope. And this is the idea of the term that I introduced at the beginning of my talk of a grommet, where you have this metal ring that is potentially stopping this tarp or this sail in this image here from fraying. Perhaps that's what the escorts are doing prior to sealing here, when we might have high tension on the nuclear envelope, for example, and you don't want those membranes to fray, right? And so this intuitively made some sense to us, but it also begged the question, what then was the purpose of this grommet? What was the functional consequence if you don't have this? Because I mentioned that the cells are viable. And when we looked at nuclear envelope integrity, it was still intact in these cells. So we wondered, could there still be a barrier to free diffusion through the nuclear envelope that wasn't dependent on the membrane? And that's the data that I'm going to show you here, is that a proteinaceous barrier, besides nuclear pore complexes, maintains mitotic nuclear compartmentalization. And here we made use of a handy tool of 1,6-hexanediol, which will disrupt weak protein interactions fairly quickly, and will disrupt the type of proteinaceous barriers that we'd be interested in looking at. And so I'm going to show you how we acquired this data here. Here, we're using a fluorescent reporter of nuclear integrity, and that is MGM4. It is a bulky construct of maltose binding proteins and GFP that is too large to passively diffuse into the nucleoplasm. And so we can see it is excluded, especially when we stain with when we tag a nuclear porin. You can see that from the overlay. And in the quantification here, you can see how the nuclear envelope integrity of our reporter does not change in control conditions. All of this makes sense. When we treat with low concentrations, about 2.5% of the hexanediol drug, we still see no leakage into the nuclear compartment. Things start to get interesting when we treat at 5% hexanediol. So in G2 cells, that is in interphase cells here, we see leakage at about 20 minutes. And you can see that here qualitatively and quantified across the population. So this presumably is as the FG nups that are primarily responsible for the nuclear pore complex's diffusion barrier are being disrupted at this concentration of the drug. So again, not so surprising. This has been shown previously. But when we looked in the same dish of cells, this asynchronous population, and we looked not just at interphase cells, but we looked at mitotic cells, we noticed something very interesting. In the same population, the mitotic cells leaked much more quickly than the G2 cells, really suggesting to us that there was a barrier to free diffusion besides nuclear pore complexes. And so I'm going to condense a lot of work into just a couple slides here. But basically, we wondered, could the spindle body itself be a barrier to diffusion, working with the membrane to maintain nuclear integrity? And so the spindle body is this large mass, this proteinaceous mass of many proteins that's been worked on by many labs. And I'm just going to, for the sake of time, show you a quick story about one protein in particular, that's PCP1, or the ortholog of pericentrin 1. And when we take advantage of a temperature-sensitive allele of PCP1, when we shift to the restrictive temperature at this time point, we don't see leakage of our reporter in this single mutant strain. However, if we cross this mutant with a CHIM7 knockout, and at the same time point, now we see extreme leakage into the nucleoplasm, and that's quantified over here. And when we did electron microscopy on this data, it wasn't just our reporter that was leaking. So again, on the left, I'm going to show you just the single allele here, the semial temperature-sensitive allele of PCP1. You can see when we look at the ultrastructure, the nuclear envelope with nuclear pore complexes, ribosomes in the cytoplasm, all looks relatively normal. When we look at the same time point of temperature shifting in the double mutant, we can see that we have extreme leakage, not just of our reporter, but of cytosolic ribosomes into the nucleoplasm here, really suggesting to us that we need this interplay between a membrane remodeling machinery, the escorts, and a proteinaceous diffusion barrier to maintain compartmentalization. And so I know I went through that story relatively quickly. This is now published. So please, I'm happy to answer questions. And if you're interested in that, check this out. But really, we feel that we uncovered here a story of a membrane remodeling machinery restricting diffusion through cooperation with a spindle body-based diffusion barrier. And if you'll indulge me for just another minute more, my lab is still very interested in this arm of research of how we maintain and seal mitotic nuclear compartments. But what we're also very interested in now is how we establish new compartments, specifically a new plasma membrane. And so one of the reasons why I absolutely love my model organism is that it does this all the time in meiosis. So Pombe sporulation, the formation of yeast gametes, is a type of cellularization. That is a multinucleate organism or a life stage of an organism going through cytoplasmic division by the de novo creation of what's called the force-form membrane in fission yeast. And so my lab is really interested in unpacking how this is created. I'm just going to give you a bit of a teaser of that. We've begun using electron tomography to reveal potentially how new membrane is added. And so I'm just going to show you play through this electron tomogram. And all I want you to take away from this is that it is a very complicated environment. So the force-form membrane is shown here in orange, making potentially hundreds of different contacts with lipid droplets nearby, vacuoles, mitochondria, ER. And so we're really interested in, is it vesicular transport? Is it lipid transport? How do we get membrane transferred to these sites? And how do we deal with this very dynamic organization of new membrane that's being shunted to this new organelle? And so with that, I just want to thank everyone in my postdoc lab who was involved with this, especially the undergraduate Linda Chen, who's now in medical school, who was very instrumental in this work and did some awesome quantification of escort subunits that I didn't have time to go into today, as well as the folks in my current lab who are now driving our new projects. And I'll take any questions that you'll have. Thank you.
Thank you very much, Nick, for the nice talk. So we are open to questions. And I think type them in the chat or in the Q&A. I thought in the chat is better. So maybe in the meantime, people start typing. I can ask you a question, Nick. So in the absence of CHIM7, you have no sealing of the nuclear envelope, but it looks like the pores seem to keep, as far as I could see from the images, a regular size, which fits more or less the size of the spindle body. So do you think there is some connection or something else that keeps this kind of pore not completely open or not getting closer to sealing?
Yeah, I don't know. I think the most easy way to think about it in my mind without any active mechanism is just that after mitosis, you no longer have as much tension on the nuclear envelope. So in an interphase state, you're able to the membrane is much more relaxed, and so it can simply adopt this natural conformation here. But it could also be more active with the anchoring of the spindle body still being in the same physical location there. And we know very well how the link complex mediates that. That could kind of keep the spindle body over this open hole. One of the things we don't really understand, which would be interesting, is there's only one hole that we see in this CHIM7 knockout, which is, of course, a stable system. So each cell cycle is the spindle body going into that same hole, as opposed to normally making a new hole in the nuclear envelope. So we don't really understand that.
Cool. Thanks, Nick. Let me see. Go.
I have a question in the meantime. In the images that you showed of the two spindle pole bodies, and then there was this panel of images where after the cells divided, it looked like it had gone from two back to one. Is that what usually happens? So in mitosis, it doubles and then one disappears?
So are you talking about the model that I showed? This?
No, right. Very early on when you showed this large panel of images showing the waves of escorts coming in.
Oh, the waves of escorts, yes.
Even
Yeah,
before this in the image itself. Sorry, just
oh.
do you actually see only one spot? Oh, sorry, you went too fast. There was two spindle pole bodies and then or two chunks of escort. Yes, here. In G2, it's down to one. Is that how it works? Are there two and does one disappear or?
Yeah, so in fission yeast, so the actual cytokinesis happens tens of minutes after mitosis. So where I'm showing you here post mitosis, this would be what would become two cells in the G1S. So each spindle body would go off into its new cell.
Ah, I see. Okay, silly question. Thanks.
No, no. I got to remember, not everyone works on yeast. They should.
So yeah, in case you have questions, you can type them in the Q&A button, not in the chat, because apparently the chat is not working, but well, it's not supposed to work, but the Q&A, yes. So I would say, yeah, if you have questions for Nick, feel free to write them in the Q&A and Nick can answer them there. And maybe now we can, thanks a lot, Nick, for the very nice talk.